All right, so like I mentioned, this is VELP Scientifica's next webinar in our webinar series. And today specifically, we're going to be looking at nitrogen and protein determination, specifically with our Dumas and Kjeldahl methods. So today's topics, we're going to go over an introduction about VELP and who we are. We're next going to go to the chemistry of nitrogen and protein determination. We'll then look at the Kjeldahl method and some of the analytical procedures that are involved with that. We'll then look at the Dumas method and analytical procedures. And finally, we'll do a lab demo where we focus specifically on our Dumas analyzer with my colleague, Irene Garcia Barrero. So the speakers for today, like I mentioned, Irene, she's a VELP product specialist at our headquarters in Italy. And myself, my name is Corey Letizio, and I am the regional sales manager for VELP based in the Midwest. So VELP Scientifica was established in 1983. We're a privately owned company ran by the same family since inception. We have about 70 employees across the world. Our headquarters and manufacturing are located in Italy, about 30 kilometers north of Milan. Now, the part that I'm a part of, VELP Scientific Inc., we are the fully owned subsidiary based in Long Island, New York, and we provide sales, service, and support to the North American market. Now, in addition to us, we also have VELP China. They were established in 2019, and they provide sales and service to the greater China market. And along with this, we have more than 300 authorized partners in over 100 countries, so we have a global presence as well. So quickly, I wanted to go over our product portfolio. First, starting with our analytical instrumentation line. As we're going to talk about today, our Kjeldahl digestion and distillation system and elemental analyzers. But in addition to this, VELP also has solvent extractors, fiber extractors, and oxidation stability reactors. Now, on the laboratory side of things, we have magnetic stirrers and hot plates. We have homogenizers, jar testers, and a variety of other laboratory equipment for your analytical and laboratory needs. So next, I wanted to go over the local service and application support. So obviously the mission of VELP is to ensure that your instrument is running at peak performance and allowing you to operate as expected in the laboratory and food production and pharma production facilities. So to do this, we have help desk and remote support that involves local people as well as people abroad. We have application support to fine-tune some of the applications for our high-end instrumentation. We have technical assistance, and this can take the form of installation and training, as well as preventative maintenance, where we have a physical service manager come on site and install and do service on your instrument. And finally, we also do calibration certification services. And one thing I always like to mention before we get going here is the VELP ERMES cloud platform. So this is our cloud-based solution that allows customers as well as VELP service representatives to access both the instrument and the data that comes from the instrument. So you're able to monitor and control the instruments in real time, 24/7 from any smart device, including a PC, tablet, or smartphone. You can receive notifications and alerts, so you have control of the analysis while you're not physically there, and you can create and share reports. And one of the best things is the benefit from enhanced service support. So like I was mentioning, some of our analytical team or service team can directly get access to your instrument to remotely service your instrument immediately if there's any issue or problems. And you'll also receive software updates with new product features as well. So now let's touch base a little bit on the chemistry of nitrogen and protein determination. So I'll go through these slides pretty quickly because we're going to go into more depth later on in this presentation. But for our Kjeldahl technique, there are going to be 4 basic steps involving sample preparation, digestion of those samples, distillation of those samples, and finally titration. Again, we'll go through all this stuff later on in this slideshow. Now for our Dumas combustion technique, we'll also go over this in more depth later, but it's 3 steps involving combustion, reduction, and detection using our thermal conductivity detector to get milligrams of nitrogen, which can then be converted to protein. So let's look at the process and some of the steps for both Kjeldahl and Dumas. So first, at the Kjeldahl instrumentation, you're going to be weighing your sample, loading the sample into the tube, and then adding all of that catalyst and sulfuric acid that you need. You'll then run the digestion program, which can last an hour, maybe two, depending on that, going into a cooling time where the instrument, or I should say the digestion block, is cooling off, and same with the samples. You then go to the distillation portion of the Kjeldahl method. Now you're able to load tubes into an autosampler if you have an autosampler, like with our VELP UDK 169, and that's going to be about 5 minutes a sample, or you can load it and run titration manually. Either way, these results are going to come in batch form based on the batch that was originally put in the digestion block. Now with our Dumas combustion analyzer, you're going to start again with weighing your sample. But as you can see, there are a lot fewer steps where you load the sample and your control into the autosampler. You run the NDA, that's our Dumas instrument, and then you get a result about 4 minutes from when you run the NDA until you get your result. But let's go into the Kjeldahl method a little bit more in depth. So these are going to be some of the analyzers that VELP offers to help with the Kjeldahl process. So after sample preparation, we have our DK series as well as our DKL series, which are digestion blocks. Now, as you can tell, you have a multitude of positions there, with the main difference being our DK series is semi-automated, when our DKL series is fully automated. And one thing I'll bring up later as well is it's extremely important during the digestion step to have acid and fume neutralization, and this can be achieved through our JP pump and SMS scrubber. Now, moving on to our UDK series, this is the distillation step of the Kjeldahl process. And as you can see, we have 5 different lines, which we'll go into more depth later on, with different levels of automations and features. And finally, we have the titration step, where you're able to do either potentiometric titration with our UDK 149 or colorimetric titration with our 159 or 169 plus that AutoKel that finally gets you nitrogen or protein determination. So again, to go in more depth with our digestion, some of the setup, you can have 6 to 20 tubes per batch. You're going to be involving catalyst tablets and anti-foam as those temperatures get pretty high. You'll be measuring your samples through nitrogen-free weighing boats. And finally, your fume neutralization system, like I mentioned, is so important to have to avoid corrosion of your fume hood and overall breakdown of instrumentation. So some of the steps that are involved in the actual digestion, like I said, is weighing in those weighing boats, adding your sample and your KJTABST catalyst tablets in there, and then you're going to run your digestion as per your application. It could be an AOCS approved method, ISO, whatever your lab is standardized running on. It's temperature and time based with different ramps in there, and then for a while you will have to let those tubes cool down after the digestion is complete. So the consumables, and specifically what VELP offers, are KJTABST. So these are catalysts that are going to actually speed up and make more efficient the mineralization of your Kjeldahl analysis. And again, these VELP KJTABST and catalyst and antifoaming tablets are very important for overall throughput in the lab. And as you can see from this slide, some of those consumables, depending on your type of product that you're wanting to do Kjeldahl analysis, you'll have different types of KJTABST, but won't go through all of them, but we can definitely help you for your application. So with these, again, we talk about the digestion blocks. With our fully automated digestion, it is going to digest for you and do all the ramps. It is going to lift up the rack and cool it for you, and then the operator would have to remove the rack and then go to the distillation phase. With our DK series, it's semi-automated, so you do a digestion. Lab technician or somebody in the laboratory would take the samples and cool them manually, and then they would again extract them just like with our automated. And like I mentioned, we have a wide variety and suite of digestion blocks to fit any needs. What's important to note on this slide is we do have 42 place blocks, but that -26 stands for the millimeters of the tube, as only 100 milliliter tubes fit on these digestion blocks, but this is widely applicable for TKN and wastewater and things of that nature. So moving on to the distillation phase of the Kjeldahl analysis, as you can see, you need a distillation unit, distilled water, and boric acid are some of the things you'll need to do a proper distillation. And a lot of that can be supported and comes standard with our UDK series. The steps of actually running the instrument are pretty simple. where you will place the digested tube in the distiller. You'll run the selected program, and then the distilled sample is transferred to the collecting flask as pictured below. And so I won't go into too much detail about our distillation unit automation table, but this shows you that depending on your analysis needs, whether it's just distillation or it also includes titration, there's a wide variety of applications that we can support, including our new 7-inch touchscreen, and more importantly, the 21 CFR Part 11 compliance, which I know is very important for our pharmaceutical companies out there doing Kjeldahl analysis. And finally, those titration options that I was mentioning, the final step of Kjeldahl analysis, we have our UDK 129 and 139 that's going to be doing manual titration. We have our 149 that's going to have that automatic external titrator. And then finally, our automatic colorimetric titrator, which is AOCS recommended, comes standard on our UDK 159 and 169. And finally, the last thing I'll mention about Kjeldahl is our V receiver. That's our formula composed of boric acid powder and a mixture of indicators that are mentioned by AOCS method. And this is a super simple and easy way to prepare a liter of receiving solution for colorimetric titration. That way you're not storing too much space and can be done very simply and easily. So now let's move on to the Dumas method. Now to start off with, VELP offers 3 different analyzers for our Dumas method. Our NDA 701 and NDA 702 are nitrogen and protein analyzers. The main difference is with our 702, it can run off both helium and argon, and with our NDA 701, it exclusively runs off helium. Now with our CN802, that's our carbon nitrogen analyzer, and it's also a dual carrier that you can swap helium for argon at any time, but that's also going to get you carbon in addition to that nitrogen analysis, and is often good for soil analysis. So now let's briefly go over the Dumas method and kind of show you the internal working of our Dumas instrument. So the first thing you'll see here is the AUTOSAMPLER up top, and that is where the sample is going to drop when it gets to the next position. That THF is our combustion furnace, and that is where the sample is going to be combusted at about 1030 degrees Celsius. And from there, it's going to move, it's going to collect all the ashes and move through the copper oxide, where it then goes to the first water trap. Now with this first water trap, it's proprietary to the VELP, and about 99% of the water in your sample is going to be removed at this step. Now the next phase is going through the reduction furnace, and that is where oxygen is removed. And then from there, you're going to go to our second water trap. Now this is a chemical water trap where about the last 1% is chemically removed from the sample, and from there you're just left with carbon and nitrogen, and from there our carbon eliminator takes that carbon out. So then finally you reach the TCD, our thermal conductivity detector, and as you can see down in that graph below, that's where the peak is going to show up for nitrogen and protein determination, and that is done through the TCD. So our standard configuration for our Dumas AUTOSAMPLERs is 1 disk that allows 30 samples at a time. Now if you are a high throughput lab, we can add 3 additional disks to get you up to 117 samples that can be ran continuously all at one time for maximum throughput in those labs, where it's something you can put the samples in and walk away. And one thing I always like to mention about our Dumas instrument is our Dumasoft software. So all of that pertinent information is in one window that's easy to use. You can create, save, and look at a database in any sort of CSV, Excel, text, etc. You can customize those reports. Now you're able to use different nitrogen standards, which is good for different samples. And then you can also be connected to VELP ERMES connectivity, which, like I mentioned, is great for the overall instrumentation. So then one thing, again, I like to mention is with our genuine consumables, so accurate and reliable results are obtained only with genuine and original VELP consumables. All of our consumables are manufactured at our headquarters in Italy. And we do offer kits that allow you to order with a single code a pre-packed solution for 1,000 analysis, 2,000, or 4,000. And as you can see from there, it's going to include things like tinfoils, copper oxide, and a hydrone, all those things that are important to run the instrument. So to wrap up the Dumas side, it's about 3 to 5 minutes per test to get your result. You're able to analyze both solid and liquid samples There is AUTOSAMPLER capacities that can go all the way up to 117. One good thing to note is you do need high purity compressed gases. For helium and argon, we do grade 5. For oxygen, it is grade 5. And then for that compressed air that moves the AUTOSAMPLER, it just has to be water and grease free. And this is something that can be taken advantage of by using our VELP ERMES cloud platform. So let's do a little bit of comparison and then a wrap up about different nitrogen and protein solutions. So the biggest thing to note with any sort of lab or food production facility is the sample type. So understanding what type of sample and then what officially approved method you need for COAs and things like that are going to determine whether Kjeldahl or Dumas are the best solution for you. As examples, we have animal feeds, meat, dairy products up here, but you are going to know your specific application better than me. So when we look around at the turnaround time for single samples, the drawback of the Kjeldahl is that all batch positions must be filled in the digestion block. So the required time for one sample is the same amount that's required for a complete batch, the idea being that there's no urgency in data retrieval. Now with our Dumas, we're able to accept single samples, and from there, we're able to test, single position test from 3 to 5 minutes at a time. And this is good for process monitoring, process optimization, and quick decision making. So for our throughput and mix, we have batch loading, we have routine single method testing, and a lot of other batch loading steps that can be included with our Kjeldahl. Again, with the Dumas, the idea being that if we want to do continuous loading and multiple matrices are accepted in the same run, it's a complete walk-away solution that will get you results in a quick period of time. You're allowed to do multiple matrices that are accepted in that same run. So the health and safety of our instruments, again, this is sometimes a drawback of the Kjeldahl. The staff can be exposed to hazardous chemicals, specifically in the digestion step, it's important to neutralize all those acids and fumes with a fume hood. Now, there is liquid hazardous waste that is generated and needs to be disposed of. Now, on the opposite side with our Dumas, there are compressed gases that are not any harm to anybody. But the drawback being calibration controls need to be run periodically on a Dumas instrument. So to wrap up, Dumas is ideal for laboratories that need quick throughput capabilities and real-time turnaround, at-line testing as an example, and Kjeldahl is ideal for laboratories with low throughput requirements. Dumas is always good to reduce workers' exposure to hazardous chemicals, but again, Kjeldahl solutions allow low to mid throughput capabilities without requiring highly trained personnel. So now let's go to the live demo at our VELP lab. I'm going to take it over to my colleague Irene.
Welcome to this part of this webinar, in which I will show you how to perform an analysis of milk powder with NDA 702. I'm Irene Garcia from Analytical Department, and this is our instrument NDA. So this is the AUTOSAMPLER. In this part, there is the combustion reactor, and then the reduction reactor. After that, we have the water trap tube, and here there is the TCD in order to determine the nitrogen quantity of the sample. This instrument is able to perform an analysis of solid, liquid, and semi-solid samples. In this case, I will perform a solid sample. It's really easy as a sample to perform an analysis. I take a tin foil, put it on the balance, and do the tara. After that, I will weigh the sample. The quantity of the sample depend on the expected nitrogen of the sample, the homogeneity of the sample, in this case is really homogeneity, and of the combustion. So we place the sample on the tin foil, close it in order to do a little ball. And after the stabilization, we can enter the wave from the balance into the software of the NDA. For liquid samples as milk, it's really simple, but first we should introduce super absorbent powder in order to absorb the liquid part of the sample, and after take the liquid under stirring. So in this case, we have already done the little ball, and we can put it on the sample holder and introduce it into the AUTOSAMPLER. This is the first position, and then the second, third, and so on, till 13 samples. Okay, and this is how to perform an analysis. Now I will show you how to start an analysis from the software. So in order to start an analysis, it's mandatory to enter the sample name. In this case is milk powder. The weight, in this case, we enter the weight from the balance. The method, is possible to introduce the method. This is the library of the standard methods. It's possible to perform or personalize some methods. The test type. For the sample test type, it's necessary to introduce the sample, the weight sample, to obtain the quantity of nitrogen. For checkups or blanks, is not necessary. And we can select the proper calibration curve. The proper calibration curve is the calibration curve in which the expected nitrogen is in the middle of the calibration. So we select with the right button of the mouse, set start. Should change it. And then start the analysis. In this case, we pair a little green arrow in order to show us that the analysis is analyzing. This is the database. So at the end of the analysis, we obtain an area, the results. For example, I analyzed this sample, and we obtain a good result of nitrogen. And this is the average of these 4 analyses. That is the standard deviation and the relative standard deviation. In this case, is really good, these results. It's possible to perform an analysis, IR report, creating this report, or even export the data in Excel, text, and LIMS. As I told you, there is a library with a lot of methods, and it's possible to perform some new methods. Even we can perform some new standards. This is the 2 standards which is coming with the software, but it's possible to perform new standards. In this right part of the database, we have all the data of the values of the instrument in this real moment. I mean, the detector, the flow of the carrier, the temperatures of the combustion reactor and the reduction reactor, the pressure, and the maintenance, because we need to know how many analyses we can perform with these consumables. After change the combustion reactor, reduction reactor as collector or water trap 2, we change the maintenance, and here the software will change. Here at the bottom, it's possible to see the analysis how is coming. In this case, the TCD knows that the nitrogen inside is flowing in this moment. It's possible to open, save, and create a new database to introduce the laboratory information to create a personalized reports. And it's possible to collocate this instrument in ERMES. ERMES is an online platform, really safe, in which you can share with your colleagues your reports, your analysis, even so how is your instrument and your analysis at the real moment. And it's possible to, in this case for the NDA, to introduce the sample weight from your personal device, for example, your tablet or your smartphone, and start or put the instrument in a standby mode, not from the instrument, as I told you, from your device or your personalized device. This analysis is finishing, and we can show the results that we have obtained. Analysis completely, and we have the same, more or less, results at the beginning. This shows us that the instrument works really properly. Thank you very much for your attention. If you need some more information or you have some doubts about your analysis or how to develop your new methods, please contact us from our website. And that's all. Thank you very much.
All right, so thank you, Irene, for that live demo. Now we are going to go towards the the questions part here. And I saw a few questions coming in, so give me a second here while I open those up. So yes, the first question, are both the Kjeldahl and Dumas techniques official techniques for nitrogen determination in milk? So the short answer is yes, they both are. They both are officially approved methods by AOCS ISO. The biggest thing, like I was mentioned previously, is just to make sure that which method you choose should be dependent on what regulatory compliances you need to have, as well as any other things for COAs that you might need. So the next question is, when the sample is liquid, what is the sample preparation procedure? Do we have to make the sample semi-liquid? So there is a way for you to solidify the sample. It is called our superabsorbent powder, and it's something that ensures that the liquid will stay completely contained in the tin foil when it's being analyzed, and it's something that comes standard with all of our systems. So the next question is, let me see here. What is the maximum sample size that can be used with the Dumas method? So we say in general, a gram is about the proper time or the proper amount, but it all depends on the type and availability of your sample. So we recommend that you check with our analytical department first. So I think that's it here. That's all the questions I see for now. Thank you all for joining us for our next installment of the VELP webinar. Again, if you have any questions or need the PowerPoint after, we can provide you with that. And I hope you all have a good rest of your day. Thank you.